MSD, I work near Philadelphia at the North Wales site in a group that does high throughput screening. And what that means is basically we have a lot of robots, a lot of cool toys, and we run a lot of biological assays. And I'm going to go into one use case of machine learning. I'm not going to try to cover everything that is done within MSD. I'll just tell you one thing that I was involved in. And so to motivate this talk, let me give you an overview, in part because this type of field might not be what some of you are used to thinking about. So it's, first I'll motivate the technology, and in this case the technology is a biological technology. And I'll tell you about the problem. So basically we're measuring gene expression in order to try to understand the effects of compounds on cells and to understand disease states. And so we're doing this by treating tens of thousands, hundreds of thousands of samples with different compounds and looking at the gene expression that's elicited by these treatments. And what we see is that you know, we'd like all of our data to be clean and perfect and reproducible, and that's not always the case. And so we have to contend with noise. And so what this story is going to be about is how we use machine learning to overcome some of the noise in this biological data. In particular, we use a metric learning approach to try to clean up the data, but then we found that by looking inside the model that was learned, looking at the internal representation, we can actually capture a good bit of biology and chemistry. And I'll give you some examples of what this model can do. So to set the stage, what we're basically trying to do is we're trying to understand, to take the pulse of this cell, right? So we're trying to understand what's going on in a cell in a disease state, or in the case where we're treating with a drug or a drug candidate, try to understand how it's reacting. And so bringing you back to perhaps further back than you'd like to remember, the central dogma of biology, where all of our cells have the same DNA in them, and what makes a skin cell different from a liver cell is what genes are expressed in that cell. So the way that genes are expressed is RNA is made, and then that RNA is translated into protein, which then gives structure and function to your body. And so if you want to take the pulse of the cell, you might think that the best thing to do is to measure the proteins, because that's the business end. But unfortunately, there isn't yet a good way to cheaply, reliably, comprehensively measure all of the proteins in a sample. And so we take one step back and look at the RNA as a proxy. And for this, there have been technologies developed since the 90s that allow you to measure all of the RNAs in a sample pretty reliably and pretty cheaply. And so this Affymetrix technology, again, is 15 years plus old, 20 years old, and allows you to look at the expression state of all the genes in the human genome. So what has changed more recently is that this process has gotten miniaturized and industrialized. And so now, instead of looking at hundreds of samples in a year, we can look at hundreds of thousands. And so the first step to miniaturize this is to realize that you don't need to measure all the genes. So we have 20,000 genes in our genome, but they're not all unique snowflakes. They actually follow certain kinds of rules. And so in this example heat map, which I hope you can see, green represents upregulated genes, red represent downregulated, and so the columns are genes and the rows are samples. And the thing to take away from this is that, you know, you can see large blocks of color. This is clustered, obviously. You see large blocks of color, which indicate that there are similarities. So a bunch of different genes are all upregulated across all these samples and then downregulated here. And this is because genes are co-regulated. They're regulated in similar fashion. And so you can extract the essence of this information by taking representative genes. Okay, and so you can reduce the 20,000 genes in the genome to about 1,000 representatives that give you all the information that's in the genome. So, you know, you guys must be familiar with other dimensionality reduction techniques, SVD, CUR, and so on and so forth, so similar approach. But this quantitative reduction in the number of genes that you measure leads to a qualitative effect on the sample throughput. So now we can reduce this from a centimeter squared array to a platform where we can have 384 samples in a plate about the size of a deck of cards. And we can use robotics to move samples back and forth, and we can increase our throughput by at least 100-fold. And so this is technology developed at the Broad Institute of Harvard and MIT, and that's being commercialized by a company called Geometry that we're working with. So what does this data look like? I'm going to show you what it looks like before I tell you what we use it for. So this is a sample, one experiment, about 7,500 samples that's been projected to 2 dimensions using t-SNE, a dimensionality reduction approach. And here the axes don't mean anything. What's important is the proximity of points. And so 2 points that are close together have similar gene expression profiles. And so what we're doing here is we're treating cells with a bunch of different compounds, and these compounds come from different classes, from different projects. So the compounds in yellow are from an antiviral program, and the compounds in red are from an anti-inflammatory program, and some of the other colors, black is third most populous, are sort of from a diversity set. And so you can see that the first thing is that the colors segregate, right? You can see yellow separating from red and largely from black. And this is because when you have compounds that have different effects on the cell, they elicit different gene expression responses, and so the cell behaves differently, and it looks different when you take its pulse using these 1,000 genes. So why would we want to do this on such a large scale, as opposed to sort of on a smaller scale the way it's been done for 20 years? There are 3 use cases. There are more, but I'm going to show you 3. So one of them is mode of action determination. So what if we have a compound, a drug candidate, or even a drug, which we know has an effect on the cell, on a disease model, on an organism, but we don't know how that effect is mediated. We don't know what its target is. Well, we can get its gene expression profile, and then we can compare it to a large bank of these gene expression profiles and see if there is another profile in our database that is similar. So if we find something that's similar in the database, we can look up what that compound is, what that target is, and then we can infer the target of our unknown. Very similar approach is looking for potential safety problems, right? It's exactly the same process. Here you take your compound profile, and then instead of comparing it to a database of profiles of activities that are desirable, you compare it to a database of undesirable profiles of toxic compounds. And then if you have a match, then that tells you that there's a problem you have to look out for. And then finally, repurposing. So this is using compounds for, or drugs for applications for diseases other than the ones for which they were first developed. And so if you have, for example, a disease state here that you know a compound is effective for, then you can see if there are other disease states, perhaps an orphan disease, something that doesn't have a big population, that doesn't have drug discovery dedicated to it, but if there's a match in the gene expression between the 2 disease states, that would suggest that perhaps this compound can be used for this novel indication. Alternatively, instead of looking for correlation, you can look for anti-correlation. So if you have an orphan disease, for example, for which you want to find a drug, you can look at your drug profiles and find profiles which are sort of the mirror image. So if these genes are upregulated in the disease, you want them to be downregulated by the drug. If these genes are downregulated, you want them to be upregulated. And so this suggests that this drug might reverse the gene signature, because the disease signature, and thus the disease process, because it has an inverse signature. So those are sort of 3 ways in which this type of gene expression data can be and is used. So what's the problem? The problem is that there's noise in the system, and the noise is sort of a collaborative effort of biology and technology. Biology is squishy and is not necessarily reproducible. We do these experiments by treating cells in culture, and cells feel different on different days. They wake up on different sides of the bed. And in addition to that, there are many steps in the biological technology, in the readout of these genes, and error propagates through them. And so the upshot of this is that when you do the same treatment on 2 different days, you get different results. They're correlated, but they're different. And when you try to do the types of analyses that I showed you on the previous slide, the guilt by association type approaches where you're trying to find a similar gene expression profile to infer similar function, well, if you can't get the same result on 2 different days, then that makes it more difficult to transfer knowledge between compounds. And so what I'm showing here is that if you take a compound and you rank by similarity all of the other profiles in an experiment, in this case 7,500 profiles, and ask where in that ranked list from most similar to least similar its biological duplicate is. This is its twin, the same treatment done on a different day. On average, it's 225th in the list. So that means that there are 223 samples which are more like your treatment than something that is actually the same treatment. So this is what we're up against. And the solution, we finally get to the machine learning. The solution that ended up working the best for us is a metric learning approach. And so the goal here is to learn a new metric on the data. So I showed you that by using Euclidean distance or correlation, we couldn't get a good match between things that should be the same. And so we taught this algorithm to learn that things that were treated the same way are the same, and things that were treated differently were different. So that's the learning problem, just predict whether 2 samples are replicates. And so it starts off pretty simply for those of you familiar with neural networks. It starts off as a simple feed-forward neural network. You feed in your samples. I said there were about 1,000 genes that we're measuring. It's actually 978. And you feed them into the network. They go through a couple of hidden layers with noisy sigmoid nonlinearities. So the goal here of the noise, we add noise before applying the nonlinearity. The goal here is being to favor saturated units, either very off or very on. And then we get to this 100 roughly binary, 100-dimensional hidden layer. And then things are somewhat different from the classic architectures. This is effectively a Siamese architecture where we split adjacent samples and we calculate a distance between them. So this is how we're learning a metric, by calculating the distance between adjacent samples. And then we combine that distance with the target. Is this a pair of replicates or not a pair of replicates? And we calculate a cost, we backpropagate that cost, and we learn the parameters of the network. So that's fine. It works pretty well. If you test it on a held-out test set, it's 97% accurate in terms of determining what's replicate and what's not. But this isn't that interesting. We did the experiment, so we know what the replicates are. But the payoff is that if you crack open this model and look at its internal representation, you can actually get so this model learned to do a better job of differentiating replicates from not. So how does it do that? Well, if you look inside, you can actually take this the activations of this hidden layer. They're almost binary, but you can make them binary by thresholding them. And so now you have a 100-bit vector that represents the information in this 1,000-dimensional continuous sample. And what I'm going to try to convince you of in the following slides is that this does an actually better job than the original data of not only representing what's replicate and what's not, but giving you information about the target of the compound structure, as well as activity across other assays and specificity versus promiscuity. So to start off with, this is the training objective, basically, to make replicates more like each other. And so I showed you that in the raw data, your replicates are approximately 225, or your replicate is approximately 225th in the list. In the barcode data, it's 24th. So that's good, but that's what the model was trained to do. What's more interesting is what else it does. So here if you treat two, if you have two compounds that have the same target, you'd expect them to be closer together. They would have a more similar effect on the cell because they hit the same target, so they should have a more similar expression profile than compounds which hit a different target. So that's your expectation. If you look in the raw data, you can barely see that. So if you look at the difference in the distance between two compounds that don't share a target versus two compounds that share a target, and look at the t-statistic, it's -1. So that sort of trends in the right direction, but it's not significant. In the barcode data, it's -43. So the samples that share a target are much closer together in the barcode space. If you look at, so you can cluster compounds by structure. So you can see that these compounds sort of have a similar structure to each other, and these compounds sort of have a similar structure to each other. This is often done in chemistry and cheminformatics. You can also cluster, obviously, these gene expression profiles using hierarchical cluster k-means. You'd expect that compounds that look the same sort of fit into the same places and into the same holes in the cell, and so have a similar effect. That would be your expectation. That's barely borne out in the raw data, but there's much more of an overlap of the clusterings by compound structure and by gene expression in the barcode space. Speeding up a little and putting all this data into one table, I've told you about the first 3 lines on this table. The fourth one is we can compare other bioassays. So measuring 1,000 genes is one bioassay, but we run a lot of other assays on these cells, and you'd expect that the effects of cells the effects of these compounds on cells or on biological processes would correlate, and they do better in the barcode space than they do in the original space. And finally, what was a little bit surprising is that you can actually predict large-scale properties of the cells better with the barcode data. So if you build a predictive model, a support vector regression model that tries to predict how promiscuous compounds are. So here you can have compounds that are very specific. They only hit one target, as indicated by the red dot here, or you can have compounds that are very promiscuous, hit lots of targets. You can do a better job of predicting selectivity versus promiscuity with this barcode data than you can in the original data space. And now the barcode data, remember, was generated only from this expression data. And it's reduced from 1,000-dimension continuous to 100-dimensional binary. So you lose a lot of information, but yet you're still able to capture this sort of high-level properties of that are inherent in the gene expression data. And so finally, all this has been retrospective validation. Can we do prospective validation? Can we actually predict something and then show that it's true? In fact, we can. I hope you can see. So this is a t-SNE plot like I showed you at the beginning of the talk, where we have lots of treatments. You can zoom in on that map to a certain spot where we know what some of the compounds do. We know that these compounds labeled in orange affect a certain target, MAP kinase. And we can ask, are the compounds in the neighborhood, or the treatments in the neighborhood of these knowns, do they have the same biology? You'd predict that they would also affect the same target. Do they? Well, in the raw data space, there are not that many in the neighborhood, and so we found 2. We tested them prospectively in an assay for MAP kinase, and we found that they're indeed active. In the barcode data, we captured those same 2, but we captured 4 additional ones, plus 2 false positives, so it's no free lunch, but we are getting more samples that are implicated or which are de-orphaned in the sense that we're finding their targets by using the barcode data. And so that's basically it. We have used metric learning to improve the reliability of the data that we're getting from these large-scale gene expression experiments by using the internal representation of the metric learning model. We've denoised the data, or alternatively, we've extracted latent factors, and we've increased the ability to pull out things related to underlying chemistry and biology. And obviously, there's the potential to apply this sort of approach to other areas where data isn't perfect and there might be some noise. And so with that, I just want to thank the organizers again and acknowledge my collaborators as well as Geometry, and make a shout out for open source software. MSD doesn't build machine learning toolkits. We take advantage of those that are available in the ecosystem. So, thank you.
Thank you very much. And we have time for a couple of questions.
Oh, thank you for the talk. I have one question. If I understand correctly, your method for measuring the mRNA is qualitative and not quantitative. Is that right?
It is quantitative. It is.
So you're able to distinguish what's the difference between the expression?
Yes.
Okay, thank you.
Any questions?
Thank you very much for the talk. Some of the people here are somehow processing, classifying, or correlating not single events but sequences of events. So for example, like the speech recognition guys, there is not one word but a word of sequence, and we want to decode it all.
Yeah.
Are there examples of some sequential classification also in your domain? For example, some temporal evolution of drag effects on some cell, or am I completely off?
No, you're not completely off there. There's a lot of time domain modeling that you can look at in terms of pharmacokinetics and things of that nature, exactly measuring drug levels and drug responses. And then in biology in general, looking, looking at the DNA strand as sort of temporal component, you can march along it, and there are lots of successes using hidden Markov models and whatever. So yes, there are definitely uses for sequence data. One of my interests is trying to apply it to, for example, chemical structures, which can also be thought of in some ways in the sequence. So, yeah.
Are you considering to combine this data with gene ontology? Because in the cells you can have the major pathway with a major protein, so are you planning to combine it? Because by distinguishing major proteins in the pathway, you can distinguish, maybe you can distinguish replicates more reliably, if you're planning to use it.
Yes, so we did benchmark that approach. So basically, the idea is not to look at single genes, but to combine genes that have a similar effect, and maybe noise in one gene would cancel out and things would reinforce. And that does better than the raw data, but not as well as the metric one.
Hi, thank you for the presentation. Last year, the company Merck hosted a competition on Kaggle for something similar. It was for a drug reception study, and the algorithm that won the competition was some deep learning framework using restricted Boltzmann machine and so on. And apparently it is implemented already by Merck. So can you please let us know some details of the algorithm that you use?
Yeah, so the specific application was to predicting function from chemical structure. So it wasn't looking at trying to decouple the behavior of the cell. It's trying to start directly from chemical structure. What can I predict from this structure in terms of what the activity of this molecule will be? And the big success from that model, which was just a deep feed-forward net, actually, it wasn't a restricted Boltzmann machine, but that showed a significant improvement in multitask learning. So for a given, if you give it a single data set at a time, it doesn't do much better than the state of the art in our in-house state of the art at the time. But you benefit from giving it many different data sets at once, and it sort of learns to transfer information between them. And so that was the big payoff.
seen biological correlates with a barcode representation? Are you treating the barcode representation just as a black box representation which is non-interpretable, or are you seeing something which relates to non-biological either aspects or...
Right. Right now we are treating it as a black box because we haven't found anything interpretable in it. I mean, we're not using only this. We're not using this in isolation. We're using this in addition to the sort of approaches I mentioned before, based on grouping genes, based on function. And so this is part of the analysis. But in terms of trying to figure out what each of those bits means, I haven't figured it out yet.
You described how you use these tests or analysis for existing materials, for existing chemicals, compounds. Do you think it would be viable to use for theoretical compounds, for example, do model molecules and then predict how they will interact in the body?
So this model is based on having the molecule in hand so you can treat cells with it and get a gene profile. But the model that was mentioned before, the one that the catalog competition was based on, which starts just from chemical structure, that you can use theoretical molecules from, because they're anything you can imagine, you can put to the model and ask it to predict how it works. So that's basically how those models, they call it Q-star quantitative structure-activity relationships. That's how those models are often used, is you then score virtual libraries and use that scoring to suggest what molecule the chemist should be using.
Hello. Did you also did you also try to learn the barcode representation not from the reduced set of 1,000 features, but directly from the 20,000 genes?
No, we haven't. So that there is less data. I mean, the nice thing about this platform is that we have a data set now of close to 100,000 samples, which we don't have in a uniform sense genome-wide. However, in the databases, there are lots of one-off experiments. People do experiments 3 or 6 samples at a time and deposit them, and so you could think about aggregating all those public domain experiments that are genome-wide to do this sort of approach, where the noise is even greater because there's lab-to-lab variability and time point importance and all that. But it's not a bad idea. It's just something that
Thank you.
Yep.
Hello, I wonder, is it important for this kind of analysis also to know the structure of the compounds that are used, and what methods do you, if so, what methods do you use to resolve this structure, if it's crystallography or NMR or some other methods?
So the structure of molecules is known because they're made internally by chemists, and they're designed to be certain molecules, and they're verified that they got the molecule that they observed. Do we need the structural information for this analysis? No, we don't, but we can combine the structural models along with the phenotype-based models and get increased reliability in terms of predicting the function. Probably predicting adverse events too, even though we haven't validated that.
I have one philosophical question. Supposing there is a drug that can enhance the human body without any side effects,
Okay.
would it be possible to find this drug using machine learning or some kind of computational optimization?
It would be possible if the problem with that is that the optimization criterion isn't very clear. But if you could specify very clearly what you wanted, you could use the types of approaches mentioned here, as well as the structure-based models to come up with a list of candidates that then you'd have to test. So this is not, it's never going to get you all the way to an effective drug. It's just going to help you look in the right place.
Okay, thank you very much.